Okay, hello to all of you. Let's start discussing chapter number 19. And chapter number 19 is basically a continuation of glycolysis. So, if you remember, we have discussed, I don't remember the exact chapter number. So, we have started the cellular respiration. And the first step in cellular respiration is glycolysis, in which glucose is converted into pyruvate. So, this is a process. Cellular respiration or aerobic respiration is a process in which the energy is released from the food we have eaten. It's called aerobic respiration because it needs oxygen. Why it's called cellular respiration? Because it's taking place in our cells. And this whole process, cellular respiration or aerobic respiration, has various stages. The first stage is glycolysis, which we have already covered. So, in this whole process of cellular respiration or aerobic respiration, we start with one molecule of glucose, and we see that one molecule of glucose, which is actually representing our food, goes through different stages, and it gets converted into different intermediate metabolites, and energy is released in every stage. And also in every stage, the high energy electrons which are there in the food, they are stripped from the food, they're taken away from the food, and they're taken by the electron carriers. One of the electron carrier is NAD plus, another is FAD. So these are the two important electron carriers involved in this process. So some of the energy is released in glycolysis, and we have already covered it's a 2 molecules of ATP and 2 molecules of NAD plus are converted into 2 molecules of NADH. And these electron carriers, they are actually getting the electrons, they are carrying the electrons, so they are getting reduced and the food is getting oxidized. So all these things we have already talked about. So reaching once the glycolysis is finished, glucose is converted into pyruvate. So what is the fate of pyruvate after that? Okay, so going from glucose to pyruvate, 2 molecules of ATP are produced, and they're produced by a process called substrate level phosphorylation that we have discussed in the last chapter. And also 2 molecules of NAD plus, they are reduced to 2 molecules of NADH, and these electron carriers, they are carrying electrons to the destination. And what is the destination of these electrons? The destination of these electrons is in the inner membrane of mitochondria. Why inner membrane of mitochondria? Because in the inner membrane of mitochondria are embedded some protein complexes, and those protein complexes are made of some proteins which have some metallic ions in them, and those electron carriers, they basically take away the electrons, and these electrons, they flow through these electron carriers. So these four protein complexes, they basically form the electron transport chain, and the end of the electron transport chain is oxygen. So this oxygen is actually pulling the electrons towards itself, because oxygen is very electronegative in nature. It's a pull of oxygen which is making these electrons flow through these proteins, and eventually oxygen gets reduced and it forms water, and on the way, by a process of oxidative phosphorylation, a lot of molecules are formed. So this is basically the contents of our chapter number 19 and 20th. So in chapter number 19, I'm going to focus what happens to pyruvate. So first of all, pyruvate will be converted into a metabolite called acetyl coenzyme A, and then this acetyl coenzyme A enters into reactions, and those reactions operate in a cyclic way. That's why it's called a cycle. And it's called a Krebs cycle on the name of the scientist who discovered this, and it is also called citric acid cycle. And you will see that there are 8 reactions in the citric acid cycle, and the glucose will be quite oxidized. The glucose will be oxidized once the acetyl coenzyme A, because acetyl coenzyme A actually represents glucose. It's an intermediate metabolite of glucose. So in this glucose will be oxidized, and some energy will be released in the form of ATP, and you will see that NAD plus and FAD, these are the two electron carriers which are involved in the reactions of citric acid cycle, and they get reduced and they will be taking electrons to the inner membrane of mitochondria. So chapter number 19 and 20th, they are connected, and these two chapters are connected to the glycolysis, which we have already studied. So this is all what is written here, and I will be talking about all these things in this chapter also. So this is basically showing you all the stages of your cellular respiration or the aerobic respiration. So the first stage is glycolysis. In glycolysis, glucose is converted into pyruvate, and in this 2 molecules of ATP are produced and 2 molecules of NAD plus gets reduced into 2 molecules of NADH. And this glycolysis takes place in the cytoplasm, and this step can take place even in the absence of oxygen. Okay, after pyruvate is formed, pyruvate is converted into acetyl coenzyme A. Okay, pyruvate has 3 carbons. Acetyl coenzyme A has basically acetyl, or acetyl word is coming from acetate. Acetyl has 2 carbons. So where the third carbon is going? The third carbon is released here as carbon dioxide. So this is the first step in this whole cellular respiration, where the first molecule of carbon dioxide is released. This is the carbon dioxide which we actually exhale. So in the process of cellular respiration, you will understand why do we inhale oxygen and why do we exhale carbon dioxide. We inhale oxygen because oxygen acts as the final electron acceptor. That's why we need oxygen, because oxygen is actually helping in the process of extraction of the energy from the food we eat. If there is no oxygen, there cannot be extraction of the energy. The energy is in the food we eat, definitely. It's in the chemical bonds, it's in the covalent bonds, which make all these macromolecules like proteins, carbohydrates and lipids. But this energy has to be extracted, and it is oxygen which it's oxygen's role to help in the extraction of this energy from the food, and oxygen acts as the final electron acceptor. And we are taking out carbon dioxide, we are exhaling carbon dioxide. It is all the byproduct of carbon dioxide, is the byproduct of cellular respiration. And here, when pyruvate is converted into acetyl coenzyme A, the first molecule of carbon dioxide is released. Now this acetyl coenzyme A enters into a cyclic 8 reactions, and they go in a cyclic way, because in these reactions we start with one compound called oxaloacetate, which has 4 carbons, and we end up with oxaloacetate. So these reactions go in a cyclic way, that's why it's called citric acid cycle. And so these two things we are going to discuss in chapter number 19, and in chapter number 20th we are going to talk about all the electron carriers, these electron carriers which have carried the electrons here in glycolysis, also in the conversion step from pyruvate to acetyl coenzyme A, also in citric acid. All these electron carriers which are basically NAD plus and FAD, and they get reduced to NADH and FADH2. So all these electrons which are being carried by them, they will be taking these electrons to the inner membrane of mitochondria, where the 4 protein complexes are embedded, and that is these 4 protein complexes, they are called electron transport chain. And then in the inner membrane of mitochondria, there is another enzyme called ATP synthase, and these electrons, they basically flow through these proteins, and they when these electrons are flowing through these proteins, they are also involved in pumping of the protons to the intermembrane space, and then these protons, because they get accumulated, or there's a high concentration of protons in the intermembrane space, then they flow back through the intermembranes. These protons, they flow back from the intermembrane space to the mitochondrial matrix through an enzyme called ATP synthase, and ATP synthase actually gets active. It becomes catalytically active, and because of some conformational changes which takes place in this enzyme, and it results in the formation of a lot of molecules of ATP. So this process of the flowing of these protons through this enzyme, this process is called chemiosmosis. So the electron transport chain and the chemiosmosis, these two things together constitute a process called oxidative phosphorylation. So electron transport chain and oxidative phosphorylation we will be concentrating on in the chapter number 20th. So this was the overall story I told you. So it's very important to understand the structure of mitochondria. I can explain the structure of mitochondria from here. So mitochondria has the outer membrane as well as it has the inner membrane. So this inner membrane, as you can see, this is invaginated. So why it is invaginated? So that the surface area can be increased. So what's the need to increase the surface area? So we have not developed the structure of mitochondria. It's there in the nature. So the inner membrane of mitochondria is invaginated to increase the surface area because there are a lot of proteins embedded in this inner membrane of mitochondria. And these proteins which are embedded in the inner membrane of mitochondria, they make the electron transport chain. So these invaginations of the inner membrane of mitochondria, they are called cristae. And this space is called mitochondrial matrix. So this is outer membrane, this is inner membrane, this part is called mitochondrial matrix. This space which is between outer and inner membrane, this is called intermembrane space. So all these terminology is very important to know because we will be talking a lot about the different locations of mitochondria in chapter number 20th. Okay, so basically, okay, let me explain from here. So as we just discussed, the pyruvate which is produced by glycolysis, it is oxidized, which means it loses more electrons, and it is converted into acetyl coenzyme A. So it is oxidized, which means it loses electrons. It also loses one molecule of carbon dioxide, which means it loses one carbon. So pyruvate gets converted into acetyl group, and acetyl group then combines with the intermediate coenzyme A, and it forms acetyl coenzyme A. So basically this is, we call it a conversion step where pyruvate is converted into acetyl coenzyme A, and this takes place with the help of the enzyme called pyruvate dehydrogenase. It's a multi-protein complex. Pyruvate dehydrogenase, it's a multi-protein complex. Okay, so this slide shows you all the reactions of citric acid cycle. I'm going to discuss this chapter with the help of this slide because this slide has everything, and I would just like to go over one more time here about this conversion step. So you can see this is pyruvate. You can see it has 3 carbons. So one of the carbon is as carbon dioxide. So this part is lost as carbon dioxide. So stripping of electrons takes place, and NAD plus is carrying these electrons, and NAD plus gets itself reduced into NADH. And the acetyl group which is remaining, it combines with coenzyme A, okay, and it forms acetyl coenzyme A. So this is what the conversion step is. So after this, pyruvate is converted into acetyl coenzyme A. It enters into these reactions. It's also given here. Pyruvate gets converted into acetyl coenzyme A. So acetyl coenzyme A, when it is formed, it combines with a compound called oxaloacetate. So the citric acid cycle, another important thing to know about citric acid cycle is citric acid cycle as well as the conversion of pyruvate to acetyl coenzyme A, both these things takes place in mitochondrial matrix. Okay, so oxaloacetate is there in the mitochondrial matrix already. So acetyl coenzyme A, which has 2 carbons, combines with a compound called oxaloacetate. So 2 carbons, 4 carbons, it forms a compound called citrate. So citrate has 6 carbons. Okay, so this takes place with the help of the enzyme called citrate synthetase. Okay, after that, citrate is converted into a compound called isocitrate. So it's just they are both citrate and isocitrate. They are isomers. And this takes place with the help of the enzyme called aconitase. Then isocitrate is then converted to alpha ketoglutarate. Now in this reaction, when isocitrate is converted into alpha ketoglutarate, first of all, there is a loss of one molecule of carbon dioxide, which means isocitrate, it has how many carbons? It has 6 carbons. So when it loses one carbon dioxide, which means it loses one carbon, it is converted into a compound called alpha ketoglutarate, and alpha ketoglutarate has 5 carbons in it. Another important thing which is taking place here is this isocitrate is getting oxidized, which means it is losing electrons. And where are those electrons going? Those electrons are accepted by this electron carrier NAD plus, and NAD plus gets converted into NADH, which means NAD plus is reduced into NADH of the enzyme called isocitrate dehydrogenase. Then this alpha ketoglutarate in the next step is converted into a compound called succinyl coenzyme A. Now alpha ketoglutarate has 5 carbons. In this step there is again loss of one molecule of carbon dioxide, which means alpha ketoglutarate has 5 carbons, it loses one molecule of carbon. So in succinyl coenzyme A there are 4 carbons. Okay, so basically acetyl coenzyme A, which is reaching to this point, is representing our food, and it has 2 carbons in it, and those 2 carbons, they have been lost as 2 molecules of carbon dioxide reaching up to here. Okay, so which means that the food we have eaten, the glucose is almost oxidized here. The carbons are actually lost. The food is still not completely oxidized, because you will see that the succinyl coenzyme A, which is now the metabolite, it's an intermediate metabolite. Okay, it will be further so alpha ketoglutarate, it loses carbon dioxide, which forms succinyl coenzyme A, and also alpha ketoglutarate also gets oxidized, and one molecule of NAD plus gets converted into NADH, reduced into NADH. Now succinyl coenzyme A in the next step is converted into succinate. Okay, and in this step, when succinyl coenzyme A gets converted into succinate, it takes place with the help of the enzyme succinyl coenzyme A synthetase. Here one molecule of ATP is also produced, and this is produced by the process of substrate level phosphorylation. So first of all, when succinyl coenzyme A is converted into succinate, GDP is converted into GTP, and then this GTP is converted into ATP. So there is one molecule of ATP produced here, and it is produced by substrate level phosphorylation. Then succinate is converted into fumarate, and this step takes place, is a very, very important step, and this takes place with the help of the enzyme succinate dehydrogenase. And here succinate is actually oxidized to fumarate. Succinate still has high energy electrons in it, so succinate is oxidized to fumarate, and there is another electron carrier involved, which is called FAD, and FAD gets reduced to FADH2. Then the next step, fumarate is converted into we can say there is no oxidation taking place. Fumarate is converted into malate, and this step takes place with the help of the enzyme fumarate hydratase, because in this reaction there is an addition of water, and then eventually malate is oxidized into oxaloacetate, and here the electron carrier involved is NAD plus. NAD plus gets converted or reduced into NADH, and this takes place with the help of the enzyme malate dehydrogenase. So basically, important thing to know here is that this citric acid cycle has to be operated 2 times. Reason why? Because if you remember, from one molecule of glucose, how many molecules of pyruvate are formed? 2. Okay, so these 2 molecules of pyruvate gets converted into 2 molecules of acetyl coenzyme A. So in one cycle, one acetyl coenzyme A enters at a time. So from 2 pyruvates, there are 2 acetyl coenzymes, they are formed, but one acetyl coenzyme A enters into this cycle one at a time. So acetyl coenzyme A combines with oxaloacetate one at a time. So this cycle has to be operated 2 times. And another important thing to know is oxaloacetate is present in mitochondrial matrix. Oxaloacetate is present in mitochondrial matrix already, and acetyl coenzyme A combines with oxaloacetate, and once all these 8 reactions takes place, again these reactions end up with oxaloacetate. So we start with oxaloacetate, we end with oxaloacetate. That's why it's called a cycle. That's why it's called a Krebs cycle, or it's called a citric acid cycle. Another important thing to know from this is what is the outcome of one Krebs cycle. The outcome of one Krebs cycle is 1 molecule of ATP, 3 molecules of NADH, and 1 molecule of FADH2. So because this cycle operates 2 times per 1 molecule of glucose, so the net or the total output is 2 molecules of ATP, 6 molecules of NADH, and 2 molecules of FADH2. And how many molecules of carbon dioxide are released in every cycle? 2 molecules of carbon dioxide. But for 1 molecule of glucose, this cycle operates 2 times. 4 molecules of carbon dioxide, they are released. So this is basically overall Krebs cycle. You can go over all these steps. It gives you a little bit more details. I have almost covered everything. Here basically this is telling you about the enzyme complex, which is called pyruvate dehydrogenase. It has a lot of, it's a enzyme complex which was it's a multi-enzyme complex. It involves multiple enzymes, and these are the names of some of the enzymes which are written here. You can go over it. And this is all it is. This is just talking in more details. You can go over them. So that's all you need to know. So the things I have already discussed with you. Let me know if you have any question.